All right, welcome back after the break, either virtually or in reality. We are really thrilled to have Marc Rothenberg to speak with us today. So Marc first of all, Marc has a CV that's one of those that would break your arm if you try to, carry it around with you. He's a professor of pediatrics, director of the Division of Allergy and Immunology, the Bunning Chair holder there, director of the Cincinnati Center for Eosinophilic Disorders. I won't go through all of his background. Let me just say he has been a pioneer, especially in the area of eosinophilic gastrointestinal disorders. He was an awardee of the E. Mead Johnson Award. He's received a NIH Merit Award. He's the associate editor of Journal of Allergy and Clinical Immunology, as well as Mucosal Immunology. Marc has been a physician, scientist, educator for many programs, including the CHRCDA K-12 program, where he was previously the training director, and he also, way back, was a trainee. So Marc has also led T32s and the KL2, and he's going to give us a career talk as soon as we get this swap displays going. So welcome, Marc Rothenberg.
Thanks. How do I swamp that? One second to do it. Okay, good. There you go. It's great having experts around in so many ways. I was a trainee and I still am a trainee. Every day I learn from a lot of different types of people, and there's a lot of messages that I have that are very similar to Catherine's. Talk about my career. First, I'm going to tell you a little bit about what I've been fortunate to accomplish in my career. Tell you a little bit about my lab, and then tell you how I got to where I am. But basically, my lab and my research, has been a long-standing interest in understanding and treating allergic diseases. And I've been fortunate to have a pretty nice operation where we have a number of different components in my lab. And one aspect of the lab is really fundamental, and I'm a strong believer in fundamental research, basic science, and a good chunk of my lab, which I always consider to be the core part of the lab, which is my main tendency is the fundamental studies, a lot of work on biochemistry, cell biology, molecular biology, and signal transduction. And more recently, really about 20 years ago, I got more interested in genetics, and that led us to develop an arm of the lab that's focused on understanding genetic inheritance of allergic diseases, actually, which is a very complex trait, and there's a lot of factors related to the environment as well. We're very interested in the genetic interaction with the environment, and we've taken a number of different approaches, primarily GWAS analyses. As a physician scientist, and I first started off really working in mice, but we've really been working over, after I developed my early career assistant professorship to do a lot of translational work. And I realized, that we can do so much in humans, and we heard some elegant talks, the last talk about non-human primate studies. There's so many experimental systems which allow us to get much closer to patients. And as a physician scientist, we're doing a lot of our work in trying to mimic diseases both in mice, but also ex vivo and translational systems. And along the way, we've developed a number of different animal models for allergic disease. So we have mice upstairs in the animal room that wheeze, as you probably know. We have them with food allergy. We have ones that have anaphylaxis, and they've been very helpful. And it turns out that we can actually use these arms of my lab to communicate with each other and to test different hypotheses, which often start out in the fundamental studies, and then we can use this to screen genetic leads, and then we can go back and see, does a genetic hit? Because really in genes, we isolate them when you get genetic risk variants. You want to know, are they functional, so we can actually go back in the lab and see if they work. And then if they reinforce each other, we bring them into the translational systems. We can knock out the gene, study their biology in a clinical setting. And if things are fortunate to reinforce and repeat and there's strong evidence, we can go into patients. And I think that, especially as physician scientists, really that's one of the greatest assets that we have here in the environment, to be able to walk right across the street, take the tunnel, and to have access to patients and then to have the Schubert Center right here on our side and to do all this clinical research. And we've been fortunate to test hypotheses in patients, and I'll show you what they've been, and to actually form the basis for therapy and improve lives, and especially in children. So along the way, this is a pathway that I'm not going to go into great detail. It's only science really that I'll show, but it's really the mechanisms that we've contributed to. And it's amazing to see how science has led to molecular and cellular understandings of things that I postured about when I was a graduate student, when I was interested actually starting off in understanding eosinophilia, the increased levels of eosinophils, and what controlled it. And back then, we didn't have any real handle on it. There were hypotheses, there were model systems, but there was no molecules. Along the way, we now have a very deep understanding, and I would say one that's clinically significant, because we understand that allergen exposure targets the innate immune system. That leads to polarization of T helper cells, which are part of the adaptive immune system. And we now understand that this leads to a very essential cell that's fascinating, and it's beautiful biology, because this one cell, adaptive T cell, CD4 positive T cell, controls many aspects of the allergic reaction, particularly because it secretes these cytokines, which were discovered along the way in the last couple of decades. So this wasn't there when I first started, but basically molecules like IL-4, which regulates IgE, IL-5, which actually now we identified, and I'll take you a little bit through that as the chief eosinophil growth factor in the body, regulating eosinophil levels, and also IL-13, which orchestrates this reaction because it targets epithelial cells which express the IL-13 receptor. And what they make, and I related to the last talk, was these CCLs 11 and CCL 26, which are part of the eotaxin family of chemokines, which I described when I was actually a postdoc. And these are eosinophil chemotactic factors. So this works in coordination, and it attracts eosinophils and regulates the accumulation of eosinophils. And along the way, we've also had some students identify other regulators of this pathway, particularly microRNAs that regulate this. And more recently, we've identified something that we think is very interesting, which is an upstream pathway, which is a complex of molecules which is part of the ripoptisone involving caspase family of molecules that recognizes allergens and then triggers the early pathway, which is related to a cytokine that induces this whole response upstream called IL-33, which also releases another one called TSLP. So it's a lot of molecules that are involved. So what's the interest? Well, actually, this led to drugs, because we identified these targets. We worked on them right here in the hospital. We did a number of studies, and this was really very gratifying work. And this is a timeline for the development of a new class of drugs that treats allergies called anti-IL-5 therapeutics, which are targeting eosinophils. And this started off actually what's very interesting to me, at least, is in 1986, there's actually preclinical work in mice. And it was basic immunology that led to identification of these protein hormones that were regulating the immune system. And this was identified as a factor that was regulating B cells, but it was later shown to also be a chief eosinophil growth factor. And then I started my PhD actually in 1989. We showed that this molecule was involved in human disease, and it was regulating human eosinophil development. And then in the 1990s, we and others had developed antibodies against IL-5, which then neutralized this, and there was a great interest in eosinophils and asthma, which took the center stage for about a decade. And then in the year 2000, what was so interesting was there was the first clinical trial in this disease with an antibody, and it failed. And there was an article that came out in Lancet, and many people came up and they said this pathway was irrelevant, that the mouse model for allergies wasn't relevant, and this was just a mouse system. But right here at Cincinnati Children's, we kept on pursuing this, and we got grants from the FDA. We actually infused the drug in patients in a phase 2A trial. It was adult studies, even though we're at a pediatric hospital. A lot of our early proof of concept studies have been in adults. And we showed that this was active. We defined the mechanism in select patients, and this actually refueled the pharmaceutical industry to become interested in this pathway. And then there was a series of studies done with more modern approaches, which is personalized approaches, understanding that all asthmatics are not the same, but if you target patients that have elevated eosinophils, which is called eosinophilic asthmatic patients, they would have a very strong response to these drugs. And in 2016, it was really the first approval of a new class of medications, which is shown down here, which are neutralizing antibodies, but also antibodies that mediate cytotoxicity against eosinophils. So these are eosinophil depleting antibodies, and this was the first new class of asthma medications in 12 years. So it really brought the allergy field to the forefront of molecular science, and now there's a number of other drugs that are biologics that are targeting these pathways. But I think there's a couple of messages in here. This is a 30-year time frame, and that's a long time. And research does take a lot of patience and dedication. It involved a lot of different collaborations. It started off in basic science, molecular mechanisms, mouse models, and it involved perseverance of my lab and the hospital's environment that allowed us to pursue things and involved grant funding. All of this was essential to finally get drug companies, which would spend hundreds of millions of dollars to do clinical trials and multiple proof of concepts, and then ultimately getting approval for the new class of drugs. So be patient with your work, be dedicated to your passions, which was, this was an example of mine, and then seek out the funding and work hard to prove your theories and don't give up, which is really one of the messages. Now today, it doesn't typically take 30 years to develop a drug. It might take 10 to 20. It doesn't, I mean, we did see the mRNA vaccines, take place in 1 year really, but that was 10 years of research before that. And we're very interested in mRNAs now, vaccines, which we're using for allergies in mice here, if anybody's interested in that. But this was another discovery that was about 15 years of research that we started here in Cincinnati Children's in my lab, which was to understand how eosinophilic esophagitis, which is a disease that we really brought to the forefront here, and that's a long story about how that happened, forming a center for eosinophilic diseases, the first in the country, and then developing murine models for the disease and targeting IL-13 in mice and showing that it was leading to transcriptional change that mimicked the disease in humans, and then doing a proof of concept study here, which I really love doing as a physician scientist, to get a hold of a drug that no one's interested in using, that no one's interested in this disease, and then infusing it in patients in a controlled trial, and we actually showed that it worked, and then that would then lead to the companies taking a strong interest in now the results of a phase 3 trial, which is positive, and anticipating approval of this drug in this calendar year. So basically, what I'm trying to show you is that we've been fortunate and blessed to have a lot of success, in clinically meaningful research here at Cincinnati Children's. So I want to take you through my background, kind of mimic some things that Catherine did and talked about, but really not as many details about the stories, but to tell you some take-home messages, to give you some advice about your own careers, and if there are any questions or anything, please, bring it up during the talk. But basically, I was born, 60 years ago in New York City, and I was raised there also by a modest family. We didn't have 9 kids in the family, but only 2. My parents were hardworking people who also didn't go to college, and they valued education and the kids' education and giving to us, my sister and I, and they encouraged. My parents were both very smart, and they didn't have the degree to prove that, but they really answered all my why questions, and I was very curious. I did ask things like, why do the birds go onto the electrical lines and not get electrocuted? And when I was in high school, I did a lot of scientific pursuits, pursued science fairs, and turned my little apartment that we lived into a chemistry lab in the bathroom, and they were supportive of all of that, and that led to my curiosity about the way the world works, and also my high school motto being to change the world and to contribute to mankind through medical research. So it goes back a long way, and that's my own personal background. But I would say also similar to you, Catherine, there was some people in high school that influenced me. I had great teachers. Of course, my parents were wonderful in terms of nurturing me, but it was really my high school swimming coach that motivated us to reach high goals. And I call him a life coach. I still keep in touch with him. And although it was just a sports teacher, but, it was very important to set the goals. He taught us that it wasn't important to necessarily win the race, but it was that you had a BTE, a best time ever, and I think medical research is similar. You just got to achieve your goals, and you got to set them hopefully high, and as long as you reach them, you have to be ready for the disappointments, losing the race, maybe not getting a grant, but sticking with it and making sure you do the best you can. So I graduated high school and subsequently went to Brandeis University, which was in Massachusetts and Boston area. And it was a great place for me because it was a small liberal arts school, but it was a very strong research institution, which is why I chose it, because it'd be private school with a lot of attention and a lot of opportunities to interact with faculty, which I majored in. And that was one of the things. I always chose the hard pathway. I'm not sure that was always good, but I did biochemistry and chemistry. Whenever there was a challenge, it was related to the high school coach, I always took the challenge on, and I did graduate summa cum laude, and so I did quite well, and then I got into the Harvard Medical School MD PhD program and pursued things which I'll talk to you about, which was to take on real big challenges there. But I'll give you some messages about that. Subsequently, got married when I was an intern, and these diagrams are my kids. All the kids. Thank God, I have 4 kids, wonderful kids, and one of them got married recently, so I have 3 grandkids, and I did a lot of research before I came here, and that's one of the messages. So many people today think that when am I going to get my R01? I did a fellowship, I did a Proctor grant, whatever. But I worked so many years in training before I came here. The PhD was 3 years, and then I did a postdoc, and I'll tell you about that. And it was intense research at the bench. And then I came here in 1996 and was recruited, and things went pretty well. So I was promoted rather quickly, and then within 5 years, I was a full professor, and then I became division director 20 years ago, which was a great challenge for me because it was basically not a division, it was a section, and a pulmonary, and then I took on the challenge to build up the division, and it's been rewarding, it's to build it up and to mentor others, but it's really the research that has been very exciting to me. And a couple of years later, we set up the Cincinnati Center for Eosinophilic Diseases, and that was because I started seeing these patients that had eosinophilic GI disease, and I never heard about it before, but there was a real unmet need, and we just had a gathering of great, clinical people here and researchers that took an interest, so we set up this center without any hospital support, but the hospital gave us the support of the environment. They never asked about having any committee approvals. We just did what we needed to do, and we did a good job at it, and they were very supportive in that regard. And then about 6 years ago, we set up a national consortium, which we run out of here, called this Consortium of Eosinophilic GI Disease Researchers, which involves 16 institutes in the U.S., and it is funded by the NIH. So, you can see I've been prolific with publications. They've been very well cited in the literature, and the lab is very active today. So in terms of my messages and my advice, how did I get here? Well, I like acronyms. My name is Marc. So I'll just try to focus on this. M. So I was thinking mentorship and training. But I think mentors is so important. And Catherine told us about her mentors. And I think a great mentor is someone that, it doesn't have to be one person. And for me, it hasn't been. It's been a series of people. But it's someone that's a role model, someone that you can really look up to. And it doesn't have to be the same thing in the same people, but there should be people that serve all these roles. Because I always ask some of my trainees, like Ashley, who's your role model? And they typically don't say me, but whoever it is, it's great to have one. And that person is an advisor, and they're typically inspiring. You should find someone that inspires you, and someone also that's generous with their time. And related to mentorship, and you need to have the training, and this is something that I emphasize greatly, because today, people want to move quickly, but it's a very competitive world. I always say if I had to start today, I probably wouldn't be where I am today. It's much harder today to get a grant, and there's a lot of other influences. But you have to put the time in. You need to invest in it. And we heard earlier from Kathy how the investment in education was a high priority for her and her family. And you need to have patience along the way. So in terms of mentors, I just want to credit them in so many ways. But I sought out in my career, and it wasn't necessarily by chance. Of course, there's always good luck. You always need to have that. But it was just to go to the highest level people that I could always find. And this was my first real mentor scientifically in college, who was the chief of biochemistry, was a father of biochemistry. And although he was an MD, he was a pure biochemist. And I said to him, he's an MD, but he didn't, he just was a researcher. I said, what, biochemistry, enzymology. So what did he want to do? He said, I want to understand how enzymes work, but all he wanted to do was physical organic chemistry. He said enzymes were too complicated. He just needs to understand how chemical reactions took place. He broke everything down to the smallest element. And he was a leader, of the generation really in biochemistry, and influenced me greatly just by watching him. And at the end of my college year, I had published my first 2 papers in very good chemistry journals. I realized how little I knew, even though I was on top of my class and I worked with this guy. I just realized from talking to him and so many other people, and it's very humbling to just realize that even though you may be on top of your game, and I think that's one of the messages in science. Even the diseases that I work on, I know more than probably anyone around me. I know hardly anything, and it's very humbling. And I'll tell you different stories about patients that, one that thanked me yesterday, and I just say to myself, why is he thanking me? He's still suffering, but we're doing the best we can. So my PhD was another theme in that area, and that was seeking out one of the best immunologists at Harvard, seeking out a professor who was very distinguished, also a grandfather of immunology, who's still a mentor today, the person in the middle, and his name is K. Frank Austin. His birthday was yesterday. He was about 92, so I called him up. But the point, and he appreciated that. But the point here also, there's a number of points. One is seek out great mentors. 2 is that keep in touch with them. So 10 years after I was here, I invited him to come, and that's Tom Boat, who was the chairperson at the time, and they interacted with Frank, and it was just great to have his advice, but also to show off, and for him to be proud, and for me to be proud also that I'd achieved, a lot here. And then when I went to, so I did a PhD, and I did college research, and I was, on top of my game pretty much, but, when you finish 5 years of clinical training, which is residency, medical school for 2 years after the PhD, and then 2 years of residency because I short-tracked, and then 1 year of clinical fellowship, 5 years, you feel like you're pretty good at what you do. I remember I was very quick at seeing patients and all of that, but I knew I wanted to go into research, so I hit the Department of Genetics at Harvard Medical School, went to the top guy there, and I was lucky. He gave me a position, Phil Leader. He was, great geneticist. I'll tell you about him in a minute. But when he came there, it was very humbling because, we had the best people around in the world working in his lab. And it's basically a clinician, after 5 years, knew very little about genetics. So it was really, eye-opening, but also one that you needed to be very dedicated and basically resilient also. We heard about resilience. You have to be personally resilient to make it in research. So Phil Leader was, really responsible for breaking the genetic code, the triple codon, and he built this equipment when he was at the NIH, and this is an exhibit that you can see about him when you go to the NIH, which was a multiplex device that would allow simultaneous filtering of chemical, reagents, and at that time it says here he was filtering 45 samples at the same time, and that was considered a big breakthrough. So he helped Rothenberg break the genetic code, and although he didn't get the the Nobel Prize in Nuremberg did. He did contribute a lot to that, and he also generated the first oncogenic mouse, the onco mouse, which was the first transgenic mouse, and he did a number of things. So what was I doing in his lab? I just wanted to learn from the best, and that was when we cloned the ataxin chemokines, which he wasn't really interested in, but he gave us latitude to do whatever we wanted really in his lab. So it was really learning from not only Phil about great things in science and history and talking about politics, but it was the people in the lab that were great mentors. Everybody had a great story, and I was just an average person in the lab learning from the other people that I've kept in close contact with. So I want to move on now from mentors in training to Marc. And these are some basic messages. Ask important questions. So whatever you're doing, you only live once. You can only do this once. Don't work on something that you don't, just because you can. Pick something that you think is important. I mean, you need to have smaller questions along the way so you can publish some papers, but pick a good question, And that's really the reason I went into allergy. It wasn't like, I don't have allergies. I didn't have a relative like you had with cancer or anything like with allergies, but it was like an open area of research. I knew when I trained and I did a hem-onc fellowship and I did an allergy fellowship that allergy was open. Everyone was going into hem-onc and cardiology, but the research in allergy, there was basically it wasn't much skin testing. There was no biologics, there was no targeted therapy. That it was a great opportunity. And also the other thing I did personally was I realized that as a love for research, I didn't want to be taking care of patients all the time that were dying. People with life-threatening problems. Allergies are important, but there's not that many people that are dying from anaphylaxis. And asthma, it is a rough disease. It was the #1 cause of admission when I first got here, but it's still relatively not as taxing as some of the other fields. So I picked allergy, and that's another message. Pick a niche area, a key area that you think you can make a contribution. And as physician scientists, I really think that we need to be very careful about this, especially as pediatricians, because you're competing against all the PhDs that go into the same study sections. We heard about some pediatric incentives, but it's tough to get a grant, and it's tough to publish a paper in a good journal. So you have to pick something that's you're going to have a niche area and make sure your pediatric training is going to be advantageous. The other thing is to appreciate. I always really, I love the job. I love the opportunity to experiment on people. I just remember so many different times I had opportunities when I first, opened the human body and I was able to see the other side of the world, and it was a privilege. Of course, it was a cadaver, but it was like an honor. It was really something to respect that, that person that donated his body and the opportunity to take care of patients and to hear their stories and the trust that they place on us. And then to work in a hospital, we're here coming to work every day to help people, so it's appreciation of that. The other A is I want to bring up is advocacy. I really strongly recommend that you consider advocacy, and there's all different elements of that. My own personal focus on advocacy has been strongly engagement with patients at all levels, not just a patient that you see, a patient that you ask about their disease, but you talk to them, you hear their ideas, and you get them to be your advocate, especially for research. The other A is academicians. I think we need to appreciate that as academicians that we have a very special place in the world and people place trust in us. Advocacy is also family, and I think your family can be your greatest advocate, and you need, of course, to develop that. And finally, is advocacy should also, I strongly encourage you to think about philanthropic advocates, which can play a major role in your research and has helped me a lot along the way. So in terms of these particular things, ask important questions. The reason I got involved in eosinophilic esophagitis was because I was sitting in my little lab right here in the R building, and I had a very small little room, and they gave it to me, and it was a really crummy lab, but I was very happy to have it. And one of the doctors, Phil Putnam, came into my little office, and he said, I want to show you something. And I said, sure, I didn't know who he was, but I heard you work on eosinophils in mice, and I have this patient, and this is a slide, and he showed me the slide under the microscope. And he's, was a GI doctor. He's still a great partner. And I said, wow, this is amazing, and it was incredible. In 1999, I never heard of this disease, but it was an opportunity. And we asked important questions about something that no one knew anything about. And we developed this system here in humans, which I show here in this slide, which is that food allergy can be avoided. You can avoid the allergens, and the disease goes into complete remission. I think Catherine, as a pathologist, knows about this, but you can be very impressed that on the right, it's completely normal. And on the left, it's very abnormal. And the point is that this is a human tissue, and it's a reversible process, and we had this, we were doing things like this in mice in the lung for experimental asthma, but to have this in patients, and to not understanding anything about it, because at the time we didn't have this reaction with food. And then we showed here through the first clinical trial that was controlled, we actually just took an asthma inhaler and we had the patient swallow it instead of inhale it, and the disease went into remission. And that was the first clinical trial that we did right here. We did that also in adults, the first one, because it was it was an important glucocorticoid study, and then we showed also that PPIs can work, but not by acid suppressive mechanisms. But anyhow, the point I'm trying to show you here is this was very interesting to me. We think it is an important question, and it's allowed us to make advances and to really achieve things in the field. In terms of patient advocacy groups, this has been absolutely essential for what we do, and I really think that partnership with patients at all levels is very critical. So we call these PAGs, and they've really shaped my research in many ways, and now we're very involved in this organization that I lead called CIGR, and the PAGs work with us at all stages of the research in terms of telling us what's important research to them, how to design trials. They help us disseminate research findings. They involved in education. We have conferences with them. They influence policy. I don't know if you know, you probably don't, but there's a National Eosinophil Awareness Week now that they helped pass in Congress, and that does mean a lot. I mean, it happens once a year now. This has led to increased funding. It certainly helped with the Rare Disease Research Consortium, and it's helped us set the research agendas. This is a picture of me with the patients. We do have them come to the lab, before COVID, once a week we had a lab tour, and it's very exciting, for them. And of course for me, I learn from everybody, including every kid that comes through, but certainly parents come up with great ideas, and it's been very dynamic and bi-directional in terms of the impact that these advocates have had. The other advocacy, as I mentioned, is philanthropy. I really think that, that you should focus on this. It's amazing, one of the greatest things when I met a philanthropist once, he was talking to my son who wants to be a millionaire, and the philanthropist told him, what the greatest day in my life was? And that my son didn't know what to say, and he said, the greatest day in my life was when I gave out my first $1 million, not earned my first $1 million. So anyhow, it's amazing to hear about that, and your patients that could be very interested in giving to you. You just need to develop that relation, and they're just as happy, if not happier, to give to you than you are to receive the money. And this is just examples of the types of funding that we've been fortunate to get from patients and over the years, which makes a big difference, because research is tough, and this is typically unrestricted funds that allow us to pursue high-risk research that we certainly wouldn't be able to do without this. So this also inspires us, and there's a number of approaches that we take to do this. But in terms of it's very important to be, to rest and relax. R&R is very important. You can't just work all the time, which I am a workaholic, but I also like to play. I'm fortunate to have a wonderful wife and a beautiful family, and we enjoy a lot of time together. We take off every week for time, and it's very important to be holistic and to consider your life outside of work too, and I think that itself has allowed me to also succeed inside work. And this is a picture of my wife. It's been very supportive, and I am a skipper. I love going on boats and taking the family out on cruises, and this is the Mediterranean. But I want to bring to your attention in terms of appreciation, this was yesterday. I mean, this kid completed one of my clinical trials. He was the second patient to complete a trial that we hope is a phase 2 investigator-initiated trial. He's from Columbus, and I mean, he's an 18-year-old high school senior, and he's a wonderful kid, but I don't have, a lot of relations with him, but it's amazing the impact that you can have, because the last time I saw him, I invited him to the lab, and he came up to the lab, and he and his mother walked around, and they were greatly influenced, but he appreciated participating in the trial. He thinks he's gotten better. It's all double blind. But this is what he gave me yesterday, and I'm just so touched. I mean, it's one of the greatest days of my career, I think, really. I mean, to give me this, it's a compass. He didn't know I was a skipper, so it's very coincidental. But these are the moments that I think we have to cherish, and that's what I mean by appreciation, to appreciate, the jobs, the opportunities we have, but also to express our appreciation to others for the opportunities that we have, especially to the patients and the people that work for us, the lab personnel that are going through their own stories. So finally, C, Marc, these are my final C's. Collaboration is very important today. It's all about team science. I couldn't do 90% of what I'm doing if it wasn't through collaborations. The community is so important. We have a great community here at CCHMC. It's a super, supportive environment. There's a huge amount of thinking going on here. I always tell people one of the greatest things, the privileges that we have, is that this is a very friendly, research, user-friendly environment. Especially for pediatricians. I mean, I can't think of a better environment to be in. And finally, communication. And communication is, in my opinion, the greatest communication is to think about publications. So many people do research and it just ends up in their lab notebook. Even if it's a small communication or journal, it's very important at your early stage of your career that you communicate to the rest of the world. I strongly believe in that. First of all, we have an obligation. Even if you're funded by the K-12, an internal grant, this is the U.S. government funds that people paid their tax money for, and if it just ends up in your notebook, it's really kind of not right, in my opinion. So, but when you do some work, communicate that. It's very important for your career. If you don't have papers, you're not going to get grants. It doesn't have to be the Science and Nature paper. Those always help, but just showing consistent publications, is very important, and I think you have an obligation to do that, but it's very important for your work. So I always talk about how can you take your work that you're doing, really every year and get something out of, in terms of the literature. So in terms of collaboration, these are some of the people that we've been fortunate to work together here. The GI people, great doctors and really come up with great ideas. They're mainly clinical people, but to have the opportunity to work with them is wonderful, and it's all synergistic and complementary, and other allergists that are part of the team, and we're fortunate to work closely with, what's become the world expert in pathology on eosinophilic diseases, and the research coordinators. And we basically use the philosophy in our patients is to provide the best care, but to learn from every patient. And I think that's also a model that you can consider, to try to do the best you can, but also to learn from every patient. And there's a number of ways to achieve that. And it doesn't mean that every patient has to go into a clinical trial either. And we certainly benefit in a huge manner from working together with so many different people here, including epidemiologists, nutritionists, psychologists, informatics, database people, and the people behind the research, including the Research Foundation administration, the chairperson of the department that's behind us, and then also fortunate to be able to communicate and benefit from geneticists that we work closely. Of course, the funding agencies, we strongly believe in releasing our data, and we have this database that we put everything in called EGIT Express, and we had a company yesterday that had all these graphs they sent me in the mail. They want to write a paper just on our public data that's over here, and this was just yesterday. So it's very important to release your data, and we're having I think an important impact from there. So this is part of our team. It's the Seeger team. This was our last meeting, and it was at the NIH, and these are people from all over the U.S. that are working together, both adult physicians and epidemiologists, and a lot of pediatricians as well. So it's all about partnership, and I hope that you guys will engage, learn something from maybe my career trajectory, and I'd be happy to talk to you after this meeting's over or during the break to go over things, and I wish you all a lot of luck. Thanks for this opportunity today, Paul.
Okay, that was an inspiring talk, so you don't get away that easily.
Available,
Tesh is going to start.
Yeah, they're all available. I mean, if you could benefit from these, just like the data that gets deposited, take them and do what you need to.
So first is a comment, actually. You showed pictures of you and Tom Board, and I swear like it's 15 years, and both you and Tom still look the same. Like, I just ran into him yesterday.
This is
so-
a sign that will never go away. Did you ever read this thing? Please do not leave chairs or tables in this area. Dr. Boat. This sign has been up here for 15 years.
Yeah.
Keep that in mind. There's the chairs, there's the table. No one listens. You
Yeah.
see that?
By both you, Jeff, and Tom, like the fact that you have been consistent for so long, and I realize the challenges myself. Like I'm transitioning from an early stage to mid stage faculty, and I realize it's getting difficult and difficult because you're competing with like bigger groups, big boys, and so how do you go about this? Like, I mean, there are many times I feel to myself like I should just give this up and like go back to clinical science, and it's something that like keeps me awake for like nights. And so
Right.
how did you do this? Like,
Right.
what is
Yeah.
the secret of the success?
These are good questions. I mean, I'm also up at night, I think academics is tough, and I'm up, regularly at night thinking about the same things because, everything we do is soft money, goes away quickly if you don't perform, and you're as good as your next paper, basically. So it's tough, but the messages that I have is, we are in a, if you're a pediatrician, I just don't think there's any better place to do research. If you're going to succeed, you're in an optimal environment here. The second thing is try to pick some advantage in some area of research where you're going to have an advantage, because I just don't see people competing against, full-time PhDs doing research, without any clinical loads and just the degree of specification that they have. It's, you certainly can, and, fortunately we do as well compete, but you want to stack the odds in your favor and pick some areas that are going to be unique, and that's why we've worked on a disease that's, we have access to the patients, and we built a database and a biorepository with 40,000 samples, so we can do things like this. But my messages are basically coming back to the MARC, it's just all these areas, and the philanthropists have allowed us to leverage this in the environment and the collaborations and the people. But I think you need to work hard. I mean, that's one of the things I can't say, it didn't happen by just being fortunate, When I was younger and I had kids in the house, of course I'd try to get home, at a decent hour, which, I did do that every day, but then hang out with my wife and family and do all my home duties. But I can tell you, when everyone went to sleep at 9, 10 o'clock at night, I was then up, up every night until like 1 in the morning, doing this stuff, and it just was a lot of work, and it still is. I mean, nothing's changed, It's a lot of work, and it's not for everyone, and that's what, like Catherine said, pick what you enjoy, pick what you want to do, and if this is what you want to do, you will succeed, but it takes a lot of willpower and hard work, but the environment here is optimal, and then, that's what I generally, that's my answer to your very good question.
Other questions for Mark?
It's a good one here. Stop is raising a hand.
I was struck because you both, as a mother myself, you both have large families. I mean, you have kids and you have kids, and how did you do all that? I'm going to ask that because I know we have a lot of young people in the audience that are sort of juggling that, especially during COVID, where it's been especially challenging.
Well, in my case, it's, I have to credit my wife, really. She's been very supportive, and it's about that partnership, and advocacy that I have from the home. But, you can't neglect your family, it takes a lot of effort too, and prioritization at the highest level. But, you also need a lot of luck and blessings and things like that. But I don't have anything more to say about that. But, we're not the only ones that did this. I think pediatrics is a great field for having a family. And, you're not alone. There's, most people, who are successful, have families. It's a lone individual that doesn't, and it can be done, and it's done over and over, and that's one of the nice things about here. It's a family-oriented city. It's it's a good place to raise families, and I think that that it doesn't, so many people think, unfortunately, a lot of the clinical fellows graduated in allergy and go into practice and make a large amount of money. They always say, I want a better family life, so they went into practice and they drop out of research, But I always say, well, academics is pretty good for your family life, because you can set your hours, except when you have clinical work. There's a lot of flexibility in things. So, I think it is good for family, Catherine, what's your secret?
And listen to your family. They'll let you know. So when my son was really little, I would play with him on Saturdays, but I always had my research papers. Pulled him when he'd be off on his own, I'd be reading. And one day he said to me, Mom, can we go play? And can you leave all those papers someplace else? So, and that's when he was like 3 years old. Right? So they will let you know. And my husband said the same thing. I need one day where I'm not responsible for anything. And so you got to give them that day.
All right, I think we should probably move to the next phase of this. Thank you so much. So that was a great, inspiring talk, both of you.